So our last speaker of this session will be Carlos Loya, a venture partner at 82VS, and today he's going to tell us about his program for advancing medicines for Charcot-Marie-Tooth disease, development of antibody oligonucleotide conjugates for peripheral nerve delivery. Thanks, Carlos.
Thank you, Megan, and it's an honor to be here with people living with CMT as well as this distinguished scientific and biotech community. I'm Carlos Loya, venture partner of 82VS, the venture studio of Alloy Therapeutics, where we focus on early stage biotech company creation by leveraging Alloy Therapeutics drug discovery platforms in antibodies and antisense, which we'll talk about here. And it's, again, a pleasure to have an opportunity to share with you our early stage work that we've been doing with the CMTRF on advancing medicines for CMT, and specifically the development of antibody oligoconjugates for peripheral nerve delivery. So, okay. Okay, great. So so precision medicines such as oligonucleotides, we believe are ideally suited for treatment of CMT. And however, the delivery of oligonucleotides presents a key challenge for the treatment of peripheral neuropathies in many important therapeutic areas, particularly because of the physiological barriers that prevent effective biodistribution of oligonucleotides to the tissue of interest, and namely the blood-nerve barrier when it comes to the peripheral nervous system, or the blood-brain barrier when it comes to the CNS. So we believe that innovations are important for the delivery of oligos to improve the specificity, efficacy, safety, and in particular, ease of administration of therapies for CMT. Okay, figuring this out. So the reason why we believe that oligonucleotides are ideally suited for treatment of CMT is because oligonucleotide medicines are well established. They're FDA approved, and it's a modality that is rapidly growing, as you can see here from the 17 FDA approvals and 9 approvals just in the last 5 years. And many of these have been driven thanks to innovations in delivery, particularly hepatic delivery with GalNAc conjugation. So we believe that these modalities poised to continue growing with innovations in delivery, as well as improvements in oligonucleotide technology. And we see antibodies as well positioned to improve the delivery of oligonucleotides in hard to reach tissues, whether it's in the peripheral nerve or in the central nervous system, which is currently served by intrathecal delivery, which is invasive. And it's not only invasive, but also it results in limited biodistribution in deeper tissues with sharp diffusion barriers, whether it's in the spinal cord, the brain, and limited distribution in the peripheral nervous system. So we see antibodies that can improve the delivery of oligonucleotides, whether they're single-stranded ASOs, double-stranded siRNA, or our next-generation technology, which we call AntiClastic antisense, which are trans-Lee cyclic structures, which I'll share a bit of that now. And Alloy Therapeutics has been working over the last 3 years in developing this next-generation antisense technology in collaboration with one of the pioneers in the antisense therapeutic field, Sudhir Agarwal, who is the inventor of some of the foundational antisense chemistries, that are FDA approved, whether it's the gapmer or splice modulators. And we see we are leveraging this foundational chemistries to build our technology because it's de-risked from a manufacturing regulatory perspective. But we know the challenges that many programs have actually encountered over the decades using this particular formats due to limited biodistribution, limited potency, or narrow safety or therapeutic windows. So we're developing this next generation antisense technology using chemical engineering, not novel esoteric chemistry, using, starting with, conventional format chemistry like the gapmer, which many of you know involves modified RNA in the 5' and 3' ends and central core that's modified DNA. And we've developed two novel formats. One we call the AntiClastic splitmer, which has it involves modified DNA on the modified RNA on the 5' end and modified DNA on the 3' end, which we've seen actually have quite dramatic improvements and enhancements in specificity and potency. But then we've also developed this AntiClastic cyclic ASOs, where we take the splitmer, add a cyclizing domain in the 5' end, which does multiple things. One, it creates this transient cyclid structure, which results in improved stability, but also masks the 5' end and prevents interaction with TLRs and other pattern recognition receptors, and actually mitigates significant pro-inflammatory responses, which could be safety liabilities. Now, we've taken our technology and benchmarked it with now over 20 clinically validated or FDA-approved ASOs. And here's some in vitro dose response data where we take actually the sequence gapmers that are FDA approved or clinically validated, and we just reformat them into our AntiClastic platform, and we consistently see 10 to 50x improvements in potency, reductions in or efficacy of knockdown of greater than 90%. And this, we believe, has the potential of meaningfully improving the therapeutic window, and we've done so with targets, across therapeutic areas. We've also taken this same approach and tested the in vitro safety and pro-inflammatory potential of our platform. And when we take FDA-approved gapmers and compare them to our AntiClastic technology and use pro-inflammatory assays like human PBMCs or mouse splenocytes, we see this consistent attenuation of pro-inflammatory responses. So what does this do? We believe that it has the potential of improving the therapeutic window of new antisense therapies. And so we're looking to continue expanding the arsenal of mechanisms of our AntiClastic antisense technology beyond RNase-mediated degradation, which we know has the potential in treating CMT types like CMT1A, but we see the potential in also leveraging other mechanisms like splicing, targeting UTRs for increased expression, or targeting long non-coding RNAs to treat other CMT types. And now moving on to the delivery platform. And we're starting with transferrin receptor 1 as a shuttle target for delivery of oligos because, well, one, transferrins are one of the primary carriers of iron to many human tissues. They're attractive because they are able to quickly internalize iron into cells, sometimes at a rate of 500 molecules per second, which is beneficial if you want to shuttle oligonucleotides into cells or other drugs. And we see that it's also well validated. It's been shown both that with other groups have demonstrated that transferrin shuttles can distribute ASOs in deep non-human primate cortex or peripheral nerve. And also it's been clinically validated to deliver oligonucleotides into human skeletal muscle. So Alloy has now developed a panel fully human transferrin antibodies using our GK transgenic mouse platform and uncover a diverse set of epitopes, affinities, and internalization rates, as you can see by this isoaffinity plot, that compare well with clinical benchmarks. And by that, I mean other TfR shuttles that have demonstrated distribution of cargo into brain or muscle tissue with good cross-reactivity between human and cyno, because we know that non-human primate cross-reactivity is important for translational development. And then we've taken, this panel of human antibodies and transferrin antibodies, done deep characterization on developability, non-transferrin competition, and have chosen 13 leads, reformatted them to monovalent Fab FCs with a silent FC mutation, and have shown actually that we have really strong affinity between human and cyno. So again, important for translation, but also nicely that we have a diversity of affinities which bind to clinically validated shuttles that for the brain or the muscle. So now we've taken this 13 leads, as we call them, and done deep characterization. I'm not going to show you all of it, but we've then used this to prioritize the leads for further antibody oligoconjugation. We've tested, in human cell assays for transferrin cell binding and see very nice comparable activity, as well as rapid in vitro internalization, which has allowed us to prioritize transferrin binders into 4 leads. And we see we have binders with high affinity that now we're moving into a diversity of affinities, high internalization, that we are now moving into production of a set of antibody oligoconjugates. And we've now also benchmarked these transferrin antibodies to clinically validated oligo shuttles. And as you can see from this in vivo PK study, where we looked at just biodistribution of our transferrin binders relative to benchmark in the brain and skeletal muscle, and we see that they compare favorably. And now we're developing a set of antibody oligo conjugates, and we're testing a set of linker chemistries as well as oligo antibody ratios to identify which ones are ideal for peripheral nerve delivery. And that's the studies that are ongoing. And we, look to take that knowledge to, collaborate along with the CMTRF and advanced an AOC for CMT1A and potentially other CMT types with these shuttles. Do not, I think it goes without saying in this room how important it is to develop medicines in CMT1A, most common inherited form of peripheral neuropathy caused by overexpression of PMP22, as we heard today. So we believe that development of AOCs targeting PMP22 with potent, efficacious antisense that could be delivered either IV, preferably subcutaneously, I think could be a important treatment for CMT1A. And well, the roadmap to I think a lead candidate antibody oligo with the CMTRF, can be seen here. We've now, as I mentioned earlier, are testing a large set of antibody oligoconjugate configurations. And we look to take this data, identify the lead transferrin binders, then linker chemistries and oligo antibody ratios that are ideal for sciatic nerve and peripheral cells, and use this to then develop lead candidates and AntiClastic ASOs targeting PMP22, and then validate them in our in vitro human cell assays, as well as in vivo humanized transferrin mice. And this is an important milestone to de-risk the program, as many of you know, because this will enable us to identify development candidates and, of course, move a medicine to non-clinical and, of course, future clinical development. So I thank, all my colleagues at 82VS and Alloy, many who are here, and the CMTRF leadership for all your work and support. Thanks.
From the online audience, does the antibody transferrin conjugate trigger an immune response in vivo?
We have not seen that.
And then I have some of my own questions. The receptors that you
And I don't
target.
believe so, just based also, just maybe just backing up, based on the validation that we've seen with other transferrin antibody platforms.
Yeah.
And also just the way that we have engineered and formatted the antibody, I think would also prevent that.
Okay. And then for any receptor that you target, does that cause internalization of the receptor? So say you want to target PMP22 on the Schwann cell, will that cause the cell to actually internalize and degrade the receptor, or?
Oh, and no, and that's why one needs to, I think, choose the right set of transferrin receptors for a specific, I guess, therapeutic approach. The goal is to, of course, not degrade transferrin. That's an important, and that's why we have formatted in the way that we have initially as a monovalent FAB-FC to prevent any clustering, internalization, and potential reticulocyte depletion.
Okay, thanks.
Okay. Very encouraging work. Quick question on your anti-clastic oligos. What type of 2' and backbone modification do you use?
Primarily 2' MOE. I think that is the primary. You can leverage any chemistry, but we like, as I mentioned, stick with the fundamentals, and 2' MOE seems to do that job and does it very well.
One thing I've always been curious about, I know there's a lot of promise with this transferrin receptor targeting approach, but if we're talking about the blood-nerve barrier, presumably you hit the transferrin receptor on the perineurium. And so that gets the ASO into that cell, but then how does it get out on the other side to the Schwann cell? Does it like kick out the antibody and the conjugated ASO, or what are your thoughts on that?
That's a great question. I think we would have to do, a thorough, characterization of the distribution of this AOCs within the peripheral nerve, between the Schwann cell, the axonal compartment, and so on. And so far, I think what we can see is, and I think some of that we will get hints of that in our initial proof of concept studies in vivo, where we're going to target like PMP22. And I think effective knockdown of PMP22 will give us that initial signal that it's going to the tissue of interest. And then, doing initial, more thorough cellular characterization of where it is and how it behaves, I think will be warranted. Yeah.
Okay. Thank you, Carlos.
Thank you.
Appreciate it. Okay, everybody, I just wanted to introduce myself. I'm Anna Combs. I'm the Vice President of Philanthropy, and I would be remiss if I didn't mention our sponsors who have made today possible, well, today through Saturday. So our lead sponsor, Novartis, Augustine, who's our scientific programming sponsor for today and tomorrow. And then we have NMD Pharma and Applied Therapeutics as our patient day sponsor. So we hope you all will stick around for that and get to meet the patients and see all the wonderful work that you're doing and how that impacts them. And I'd also like to thank 82VS, that's a venture studio of Alloy Therapeutics, Alesta, Actiobia, and Takeda. And now on to some housekeeping. So we are going to take a short break. We're going to come back and be in the room and start promptly at 4:30. And for those of you that are virtual, we will see you back in that space at 4:30 as well. Okay, thank you.